Ryan, it's great to have you back on the podcast.
Yeah, great to be back. Thanks for having me again.
Yeah, and today we're going to jump in where we left off about epigenetic methylation, biological age, longevity, and this emerging science that's so fascinating these days.
Yeah. No, I'm excited to come back. I know that we didn't talk too long ago, but this field moves very, very quickly, and already a lot more to talk about.
Well, let's start off today with the unusual or non-negotiables you've done for your health, performance, and bioharmony.
Yeah, no, interesting. Bioharmony, I guess, is that new term added there, which I think that when we last talked, I was sort of talking about some of the things I've seen through my own epigenetic clock through retesting, things like stress management, among others. I think that those are still things that I'm doing. I think that probably the newest additions since we've talked have been maybe experimenting a little bit more with SGLT2 inhibitors, which can help reduce that glucose in addition to some fasting, doing some mTOR inhibition with rapamycin. Lastly, experimenting a little bit with 17 alpha estradiol for longevity as well.
What's that last one?
So it's an estrogen compound. It's an FDA-approved drug. In some of the newest trials, particularly these interventional treatment protocols, ITP program from Michigan, they've showed some longevity-related benefits. And so this is a very new one for me. I have literally just started it a little under a week ago. But I thought it was relevant to bring up. And it looks like one of those very few compounds that in that program extended the longevity in mice.
Mm-hmm. Well, this is a really pertinent question then. When you're adding these new things, sometimes they're more experimental, they're newer, they're cutting edge. How do you know what's working and what you want to keep? Like, how do you evaluate these things personally? Because it's one thing to read a paper about them and say, oh, this looks really promising in mice. And it's another to say, okay, this is actually making a difference in my life.
The one thing I have learned is never make more than one intervention at the same time. Keep it very, very controlled within an interpersonal basis. I adopt more interventions than most, and so now trying to take a lot of as much as objective measurements as possible, whether it be wearable data, sleep data, blood labs, and obviously the epigenetic testing. But I think that it also is sometimes a bet. For instance, with the rapamycin, I don't know that I necessarily feel anything one way or another, but I'm sort of betting it on based on the literature. I try and establish these protocols for a good amount of time as well. Usually I would say a minimum of 3 months as a standalone intervention before I would go on to any others.
Yeah. So we did our episode together back in April of this year, episode number 47. And you mentioned your background and your backstory. But for those who missed the episode, can you give a summary of who you are and how you got here?
Yeah, certainly. And I'll try and make it as brief as possible. But my background was in biochemistry originally, sort of working on the chemical synthesis of peptides and proteins. I used that to sort of go to medical school. Passed my USMLE Step 1 in medical school, got to my third year, but did not like it, and so made a very radical decision to pursue something else, and ended up opening a compounding pharmacy here in Kentucky around 3 months after that. And that was a compounding pharmacy that really specialized in peptide and proteins as pharmaceuticals, really new and innovative products. And we just sort of hit a niche. We grew pretty rapidly, particularly serving this sort of cash pay medicine space. A lot of people, a lot of physicians who were really interested in preventative healthcare, things like longevity, treating age as a primary outcome. And ended up selling that business in 2020 to create TruDiagnostic. And TruDiagnostic is the company that I work for now. And we specialize in epigenetic methylation, sort of health analysis, reading those epigenetic marks of gene expression in ways that might influence or inform us about your own health. And so it's a very new but growing field, and really excited to be there. We've built probably the largest private epigenetic database. I think when we last talked, we were right around 10,000 samples. Now I think we're over 16 or 17,000. So, very much scaling that out, and then trying to work with our consumers and physicians to learn as much as we can about those patterns.
Okay, we've now at this point said the word epigenetic more than once or twice. So I think we need to step back and define what epigenetics is and how that relates to biological aging, which is another term that we should probably define.
Certainly, I'll try and do both. But epigenetics is sort of above the genome. I think a lot of people are familiar with genetic testing, looking at SNPs and that coding or the hardware of your body. The epigenetics are sort of the on or off switches for that DNA. And I think the way I explained it previously, and the way I'll try and explain it again, is that these epigenetic marks regulate how your genes actually behave. So every cell in your body has the same exact DNA expression, but obviously your heart is very different than your skin, and that's due to what genes are expressed and turned on and turned off. And that's sort of what we're measuring. But particularly, we're measuring here sort of the off switch, which is called DNA methylation, just a small chemical tag that is put on to sort of silence gene transcription. We've got a lot of these in every cell, and every cell is different, over 26 to 29 million, depending on who you ask. And every cell can be widely different. And so this provides a lot of data. And this data has been used very recently in a lot of ways, but really it was brought to prevalence with this idea of testing someone's age, the difference between chronological and biological age. So chronological age, as most people know, is sort of your birthday, right? The number of years you've sort of trips you've gone around the sun. This biological aging process is sort of trying to measure the underlying physical function or biological parameters of why you're aging. And so we all know this difference in people who look 70 even though they're 50, and vice versa, people who look 50 even though they're 70. And that's really what we're trying to measure, is that how your body is performing from an aging perspective.
Hmm. And then when you drill into that, there's a bunch of different forms of biological age testing. There's telomeres, there's blood analysis algorithms, there's glycans, there's epigenetic methylation, and there's other like proxies.
Yeah, certainly. And this brings up a difficult question, which is, what is aging and how do we measure it? And this can be a hard question to answer, but I think that this is probably helpful in terms of diving into this testing and looking at what measurements are the most helpful in terms of trying to answer the question of how am I aging and what does that mean for my long-term outcomes. And so, you can start with a lot of the blood-based biomarkers and things that we know from, many decades of data that are associated with age. So we have these patterns in our blood, which are associated with age. And even before that, people were saying biological age is, your chronological age plus 1 year for every pack you smoke. So these things have continually gotten more and more sophisticated. A couple of years ago, in the early 2010s, things like telomere testing were probably considered some of the most advanced, which, telomere attrition, those end caps of your DNA, which shorten over time, those are, sort of a hallmark of aging. With that being said, there are a lot of these different patterns. So glycans are, sort of these glycosylated, sort of immunoglobulin molecules, which sort of get sugar attached to them over time, which can be indicative of certain aging processes. But whenever you're looking at the entirety of these things, I think that it all comes back to this one big idea, which is why measure age in the first place. And the reason being is that age is the number one risk factor for almost every chronic disease and death. So if you think of Alzheimer's, if you think of osteoarthritis, if you think of cancer, the biggest risk factor for the majority of those diseases is age, and not just by, I would say, a small margin. We always say that there's a reason you see people in their 60s and 70s getting a lot more disease than you would in your 30s, even though they might have the exact same risk factors. That big sort of question mark, what's making that big difference is age and the aging process itself. And so we know the aging process is made up of a lot of biological functions. In fact, biological age itself is sort of defined as this progressive loss of function as we age. And so the question is, how do we define it? And that is always expanded. As we get more and more markers and more resolution in how we can test these things will continue to improve. But right now, I would say there are two really good ways to test biological age metrics. And that is, one is how precise are they? If you take the exact same sample and retest, do you get values which are almost exactly identical or very, very similar? That's obviously important because right, if you're not getting that precision, how do you know if your testing is telling you anything really about your actual biology? And so I would say precision is a very important part. I think the other part that is incredibly important are the prediction to outcomes, right? The reason we're testing in the first place is to maybe, find out where we're going and then hopefully modify some of these factors so that we can improve that trajectory. And the way that we measure sort of that the predictive nature of these things are through hazard ratios, which is basically how likely are you to get a certain outcome if you were to see a change in this metric. And these hazard ratios are incredibly important because they sort of show you what you're measuring and how much of a difference it makes in your own life. And so for anyone who's trying to validate these things, those two things, precision, which is usually measured by a value we call the interclass correlation. And you really, the thing is you want that to be as close as one as possible so that you're getting the exact same measurement when you retest the same sample. And then secondarily to that, a hazard ratio, which is sort of I would say defined as a value that is usually 1 point something, 3.4 to 6.4. And that value after the 1 is really the percentage increase of the odds of a certain event happening. So if it's a 1.64, that means that you would have 64% increased risk of that outcome happening versus, sort of that control. And so those are two values to be very familiar with as you're judging the many, many different options of biological age testing out there.
Mm. And then specifically for epigenetic methylation, why did you guys choose to focus on that? And how do you go about identifying and comparing epigenetic methylation tests of biological age?
We got into it mainly at first because it was probably the most robust and predictive test. Even way before we got into it, people like Steve Horvath at UCLA were creating really great algorithms like BioAge and the Horvath pan tissue clock. We know that those clocks were really exciting because they had better hazard ratio prediction than almost anything out there and across the board to these age-related diseases. So they were able to predict mortality better than most things. They were able to predict cancer risk. They were able to predict things like neurodegenerative disease. And so we were measuring those all through those hazard ratios. And so that's sort of why we got into it. And we saw the potential and we saw that, the potential to also change these things. So, whenever we first got into it in 2020, we were sort of following a very important trial we talked about last time, which was published in late 2019 and really was the first trial to show a reversal of these age markers with an interventional treatment, which was metformin, growth hormone, and DHEA. So we sort of got into it the moment I think that we saw the connections, but in addition to that, the moment we could see a change. And so since then, there are a lot of algorithms which have been created and a lot of other private companies who have gotten to this space to do this type of work. And one of our favorite things to do is to try and educate on how you vet these therapies and have sort of I would say, a running list, which we can maybe go through one by one to really dive in there. But the first thing I would say is, more than anything, the algorithm should be published. And this is a tricky part because I always liken it to going to a fortune teller. You can go to a fortune teller and you can believe what they say, but you're choosing to believe it. And I think a lot of times, whenever we're talking about things like biological aging and hard science, you want it to be different. You don't want to have to just believe it. You want scientific evidence, right? And so I think that number one of which I would recommend that the algorithms are published. In those publications, there are a couple things to look for. And, as I mentioned, one is the accuracy or those ICC values. Another one is called the mean absolute error. And so this is, how much of an error range there is with these different types of testing. And then the last one is going to be those hazard ratios to disease, the connection between those outcomes. And again, I don't want to go too far, as I know you might have some questions on that, but I also think how these clocks are trained is incredibly, incredibly important and probably the most important thing to talk about.
Okay. And you mentioned in earlier, before we started recording, that there's a difference between what you called first versus second versus third generation clocks.
Certainly. And this is exactly what I'm talking about in terms of how these clocks are trained. And this is incredibly important because the first clocks, particularly even the Horvath 2013 clock, which really made this type of testing very popular, they were trained to predict the chronological age of an individual. So they took these methylation CPGs, those locations that we're testing, and they trained mathematical algorithms to predict that with the most accuracy and the least amount of error as possible. And so they were trained to predict the chronological age. And what they saw was that even though it was trained to predict chronological age, I should say that if someone was older than their predicted, their biological age, or this measurement was older than their chronological age, they were at increased risk of negative health outcomes. And vice versa, if they were younger with this testing than their chronological age, they were protected from some of those negative health outcomes. And so even though it was trained to predict a chronological age, the end result was really a biological measurement. It was measuring something in the body that was, having this type of effect on the aging processes. Those clocks continued to advance, but there were problems with those clocks. Number one of which is that the better that those clocks got, the closer they got to predicting your birth date, which for two reasons is not something we want to look at. One, we could just ask you. The second is that it's really closer to a chronological process than measuring a biological one. And this has created, I would say, quite a few issues. One of the issues that we typically see here is how it responds to treatments that we know beneficially affect the biology of aging. So for instance, Dr. Belsky from Columbia took the data. So he created one of our favorite algorithms, the Dunedin PACE, which is a third generation algorithm. Well, I think we'll get to that. But in that first generation testing, what he saw is that in a study called the calorie study, which was right around an 11% caloric restriction study, those clocks behaved a little bit abnormally. So, we all know caloric restrictions are a very well-validated therapy for health span and lifespan extension in a variety of mammals. Just about every mammal has seen some benefits of caloric restriction in some of these studies, and including humans. And in this particular study, though, those chronological clocks actually went up with caloric restriction. So they went in a way we wouldn't have expected. However, as we get into those other clocks, those newer and what we call these second and third generation clocks, they behaved exactly like we would expect. And we actually see this as well with other trends like rapamycin, another one that has, almost universally seen better results in almost all mammal species. And so chronological leaves some things to be desired. And so in addition to looking at the mean absolute error, the precision with the interclass correlation and hazard ratios, the other thing you want to look at is how that clock is trained. Is it trained to chronological age, or is it trained like a second generation clock, which are trained to more biological phenotypes, things like those blood-based biomarkers, things like physical function measurements, things like telomere length, for instance. And I think that those second-generation clocks are definitively where the field is going. I think that no one is really, focusing on those chronologically aged clocks anymore. They're a little outdated, and really the only use case now is in things like forensic testing. And so there are still people who are doing this. Even, Dr. Horvath and Riccardo Marioni just recently published a really great chronological update clock from 24,000 samples from Generation Scotland. So we're seeing these improvements, but really the application is not in a healthcare setting. It's not necessarily as much for longevity. It's just sort of to tell how old someone is if you have their DNA.
I was doing some research and I came across something called the imprintome and the Human Imprintome Project. Will you break down what that is?
Yeah, definitely. So that's an area that we are really excited about. And it's a little bit of departure from these biological age measurements, but it comes back to this idea that we inherit more from our parents than just our genetics. We actually inherit even epigenetic features. So there's a study that I recently learned about that I like to always bring up here, which is that in mice, they did a study where they exposed them to cherries. And then every time they exposed them to the smell of cherries, they sort of, had this negative sort of pain sort of response. And they did this over and over and over again. But then what they found is several generations later, the descendants of that same mouse, even though they had no conditional training themselves, still, whenever they smelled cherries, would expect pain. And so the question is, how do they inherit that type of behavior? How is that passed down? And one of the biggest theories and leading theories on how that is passed down is through sort of not just inheriting the DNA sequence, but also these epigenetic marks, which regulate gene expression and might, encode for things like behaviors. So, for instance, whenever ducks are born, they imprint on the first thing that they look at, and maybe some of those behaviors are mediated by this epigenetics. This is always a really exciting field because of some of the things that we can find out from these transgenerational inheritance. We can find and see things like markers of the Holocaust or the Rwandan genocide, for instance, in descendants. And that might impact our health. That might impact our health in ways that it's hard to control. But in a certain subset of those passed on genes, they only come from a single parent of origin. So you would only inherit that methylation from your mother, or you might only inherit that from your father. And the problem with that is that, much like genetics, if you inherit two alleles, right? And so if one allele has a problem, you can really cover it sometimes with the other allele, and it's not as much of a problem. But with these epigenetics, if you're inheriting some deficiency epigenetically from only one parent, it's much more likely to penetrate to disease. And so Randy Girdle is from Duke. He created another famous experiment out there where he took two genetically identical mice who were pregnant. And while pregnant, he fed one of the genetically identical mice methylation cofactors like vitamin B12 and folic acid. And then the other, he fed them nothing but a chemical BPA, which many people might know from things like water bottles or some of those other things. And the descendants of those mice were wildly different. The one that was fed BPA was obese. It had diabetes. It had lower muscle mass. I would say, yellow. And so if you look at these mice back to back, the other one is completely normal. The one that was fed these sort of methylation cofactors. And so the idea is that and I apologize for the long-winded explanation on a lot of these, but with that being said, what's happening here is that we know that the behaviors of our parents, particularly as we're, prior to us being born and as we're being born, give us these methylation signals which are passed along. And unfortunately, these methylation signals are really unchangeable. They really stay the same way throughout all of our cells, and they stay the same way throughout life. And so these can be huge risk factors, more similar to genetics, actually, than typical epigenetics. And so what we're really trying to do is to find those regions first. That was the first goal, to know where to look and what's imprinted. And then the second is to find out how they're affecting disease. And the idea here is that we believe that it's affecting almost every disease, everything from obesity, but especially in neurodegenerative diseases. And so we're starting to look at these as maybe features or things that can explain things like the racial differences in Alzheimer's. We don't know why that, African Americans, get Alzheimer's much more than Caucasians. It doesn't seem to be genetic. It doesn't seem to be in their blood biomarkers. But we have some preliminary data that suggests it's because of this imprintome. And so we can find out what behaviors prior to pregnancy are influencing some of these factors. We might be able to change them and prevent them from even occurring.
Have you heard of Mark Woolen in his book called It Didn't Start With You? He's like a therapist that was given a lot of cases that no one else could solve. And he traced things back to traumas their parents or their grandparents experienced that were presenting themselves in their own lives. And so it sounds like he was.
Yeah, definitely. That sounds exactly right. I mentioned, those markers of trauma we can see now at certain locations, and so I would say that that's almost certainly what he was measuring in the phenotype, right? What actually happens to these individuals, and now what we're trying to do is associate those phenotypes to those individual biological markers, which might tell us about how it's happening.
Yeah. And will those be consistent across people?
Yeah. So that's a good question, and we're even doing studies right now on identical twins to see if there are even the same among twins who are, growing up in those same conditions and are genetically identical. But we know that the imprintome regions do look to be similar among individuals. So it looks like this was evolutionarily evolved to make sure that there were certain regions which could do the type of programming to pass along fear behavior, to pass along, nutrient response behaviors, right? So if you grow up, if your parents and grandparents and many generations before you have had difficulty getting the proper nutrition, then your body wants to be encoded from that from the beginning to make sure you can respond to that. I think the answer is the same regions are yes, but really until the summer of this year, 2022, there had been the full list of these regions had never been created. And so that's really what we started to do with North Carolina State and Dr. Girdle, Dr. Hoyo, and Dr. Scar. And we sort of leveraged a lot of their work looking at eggs, looking at sperm, and then looking at ultimately the embryo to see what was fully methylated from the mother, what was fully methylated from the father, and where those regions were. And so we did that, and prior to us doing that, there were 28 of these regions known. We sort of identified 1,488 regions, and so it's a lot larger than I think we previously anticipated. And we created now the first way to do the testing of these locations in a really cost-effective manner. And so now that we know these locations, now that we have a way to really, I would say, not cheaply, but less expensively test them. The next idea is let's figure out how they relate to disease. And I think that the first types of investigations we're really excited about are going to be Alzheimer's and autism.
Mm. I love the idea of this. And the very next thing is I'm wondering, is this going to be fatalistic, or are there things that we can do after we know these, like say take methylated vitamins or avoid BPA so that we don't like succumb to the same things that our parents or grandparents or somewhere down our lineage did?
Yeah, and it's hard to say. I think that that's one of our biggest issues with genetics, right? Is sometimes you can act on it, but you can't change it, right? You can help influence your guide or guide your behaviors, and I think unfortunately that's how we probably see these now because they're really meant to be unchangeable across multiple tissues and across longitudinal changes. And so, but again, hopefully what we can do is find out what pathways, for instance, what proteins these might be making that we can have an effect on and sort of stop disease. So I think that right now we won't be able to change that specific imprintome, but we might be able to change the lifestyle, which mitigates the negative effects of the imprintome. And so we can still see benefit, I think, in the future. And ultimately, I think that the best idea would be how do we prevent these imprintome regions from being passed down? How do we make sure that prior to conception, we're doing the diet, nutrition, and the exercise and all these lifestyle factors, which we know are going to help. And one of my favorite examples of this is work that is happening at the University of Washington as it relates to pesticides. So glyphosate, is a really common pesticide that has been really cleared as safe by the FDA. The problem is, and I would say the data that the FDA has gotten is from a single generation. If you or I are exposed to this chemical and we have no negative outcomes, that's a good start. But what we're seeing, particularly with Dr. Skinner at the University of Washington, is he's seeing that actually exposure is actually changing those imprintome regions. And that we're not necessarily having any negative effects ourselves, but we're passing those effects down. And actually, for this particular chemical, Dr. Skinner doesn't see any change in even the first offspring, but he sees it in the second. What we're starting to this concept of things like genetically modified foods, which might be treated with pesticides and survive. Those pesticides might be getting into our food and might not have an effect on us, but they might have an effect on our grandchildren. And we might not be able to see those effects because we just haven't had enough time. And so I think that this research is going to be paramount as we continue to I would say, get exposed with a lot of more chemicals in our world. And we might not be seeing all those effects yet, but they might be becoming more and more prevalent. And so I think that more investigation into this and to how it's affecting future generations is also going to be very important.
Yeah, I just recently released an episode and we talked a bit about glyphosate and the way it's applied to foods and how that leads to complications, possibly in future generations also, but even immediately to your gut microbiome and how that changes the synthesis of like important biomolecules and all kinds of things that they might not have reviewed initially because they didn't recognize effects it was having outside of like your normal human cells. But I think it's smart with a lot of these things to take the precautionary principle and before spreading billions of pounds of a certain chemical, make sure that we really understand the ins and outs of it.
Exactly. And quite frankly, we're not sure if what we might be measuring in the imprintome might be a result of the microbiome changes, right? Until we have a really good comprehensive knowledge of these things, maybe scale of, distributing this to, billions of people might not be the best outcome or idea. And I think that you don't have to be a conspiracy theorist. You just have to say, we maybe don't have all the information and maybe let's go with things that we've been doing for thousands of years, which is more natural biology. And so I think that, not necessarily knocking, genetically modified foods, but I think we're starting to see that there are effects that we didn't quite intend when doing this at large scale.
Absolutely. And to be clear, measuring your biological age via epigenetics is not going to lead to any of these risks because you're not inserting anything into your body.
Yeah, definitely. And I think that it brings up this larger idea of, why is epigenetics important? And, what is it? And I think that what it is sort of a reaction to your whole system. Everything that you do, what you eat, what you, what medications you take, how you sleep, your lifestyle, even what we can see emotional regulation has an impact on these things. And so if all of those things are changing these things, the idea is that maybe they can changing them in certain ways, which we can be able to interpret and then provide a lot more context about what's happening in our health. Predict disease, predict how you might respond to medications, predict what's going to be the best way to you get better sleep. And so this is really just a very new area of investigation. But a lot of those questions, much like genetics have done over the past 30 years, will start to be done with epigenetics. And with that being said, I think this idea of biological age has taken the forefront because, again, it's the number one risk factor for all chronic disease and death. And so, if you're going to measure one thing to be preventative about your own healthcare, I think a lot of people might argue that this is the one thing to measure.
Yeah. Ryan, a few minutes ago, you mentioned an observation of twins. Why are twins important in this line of research?
Oh, for lots of reasons. I think that one of the other studies we're doing is with Stanford, and we're looking at twins who are doing two different types of diet. So, for instance, one is being put on a carnivore diet, the other one is putting on a vegan diet over the course of a few weeks. And one of the reasons we love that study is because sometimes genetics influences your epigenetics. Right? They're not always mutually exclusive. And so, by doing a study in people who have the same exact genetics, we can remove that confounding variable. And so we can get a much better idea of what is happening from a methylation regulation perspective from certain diets. And so we're really excited to do that. We're just now extending that cohort to get a little bit more data, which we're really excited about. I think it'll actually be a little bit of a follow-up to the Game Changers documentary that was on Netflix. People might be familiar with. So I think it'll be sort of released along with that. But we're really excited about it. It's probably the largest twin study that we've done. In case of the imprintome, I think that the idea is we're trying to find more about the processes, right? If everything else is the same, right? The same genetics, the same timing, then maybe there are certain things we can find out that might lead us to find out more about how these imprintome regions are actually decided and passed along. And if we can find out that, we can maybe find out ways to prevent that process in certain areas or to encourage it in certain areas. Really, twins are great because they remove confounding variables.
Yeah. Cool. Well, what else is coming down the pike in the world of longevity and biological age testing and all of this? Because you're very involved and I feel like you're on the forefront of all of this.
Yeah, we try to. I think that for anybody who might be out there, listening, we do a ton of research, and we're always in need of volunteers to measure some of these things, as well as I would say collaborators if you're doing any type of research yourself. And so we've done a lot of collaboration with a lot of different universities, and we're about to publish a few papers that I'm really, really excited about. One of the papers that I'm really excited about is something we briefly talked about last time. We've been working on it now for almost 2.5 years, which is a joint project with Harvard that we've been doing. We're trying to create the best-in-class biological age clock and by a wide margin. And the way that we're doing that is we've talked about some of these changes which happen with age, changes which happen with blood, changes which happen with telomeres, would happen with different protein markers. The better resolution you have on multiple processes, the more insight and the more accuracy you can have. I think a good example of this is the Human Genome Project. I think a lot of people, whenever it came out, thought this will be the solution to all of our problems, right? We'll know how all of these things are encoded and this information is transmitted. But then we started to see that, from genetics, you go to epigenetics, right? How those are regulated. From there, you go to transcriptomics, which is how those mRNAs created. There are multiple types of mRNAs. Then you go to, how those proteins and peptides are all created and how single nucleotide polymorphisms can change function of those products. Then going to those metabolites. And so genetics was only a small piece of this really large picture. And so what we're trying to do is to take, as much as we can, the largest picture we can. And that is to do full genome sequencing, do epigenetic sequencing that we're doing at our lab, to do proteomic, untargeted proteomic sequencing, where we're looking at over 3,500 different proteins, and then doing metabolomic sequencing, looking at the metabolites of the body out of all those processes, and then ultimately looking at the phenotypes, what is actually happening to these individuals. And we're doing that across over 50 years from these patients who have had their DNA banked, and a lot of their specimens banked in this partner's biobank at Harvard. And so, what we're trying to do is get that large picture, use all of those variables to create what we call this multiomic clock. So it'll include over 75 different blood labs. It'll include full epigenetic, full genetic, really large scale metabolomic and proteomic. So we can put all of those factors together and find out what is the biggest driver and how can we predict these negative outcomes. And so it's been a lot of money, a lot of time. We have the most amazing collaborator, Dr. Jessica Lasky-Sue at the Channing Lab at Harvard. And we're about to start publishing some of that data. So the first thing that we'll do is a chronological age clock. We know we already talked about, some of those clocks and how we're not big fans of them. So the question is, why are we publishing them in the first place? But what we're doing is I'd say, publishing methods. We're sort of showing people how to improve chronological age prediction through certain features or certain insights that we found. And one insight that we found to be very, very important is the impact of the immune system. And so, I think you and I referenced this a little bit before the call. There are a lot of people out there who are now starting to say the narrative of epigenetic age might be too related to immune system function, particularly because we're measuring blood. And in the blood, the DNA that we're measuring is from all of these immune cells. The question is, how do we make sure that changes in immune function are not giving us changes in that biological age if it's not accurate, right? If it's not predicting changes in negative outcomes. And so one of the things that we've done, and one of the other papers we're publishing with a collaborator of ours, Andrew Tischendorf, who's a genius bioinformaticist who is working in Shanghai. And we've created a way to tell you over different what the percentage of 12 different immune cells are just from that DNA methylation. So we can tell you your CD4 cells, your exhausted versus naive CD4 cells, your basophils, your neutrophils. And by controlling for those changes, we can make much more accurate predictors. And so, for instance, I mentioned a study earlier, a chronological age predictor from Dr. Marioni and Dr. Steve Horvath. Their mean absolute error set a record from those 24,000 patients. It was only 2.3 years would be I would say the most error that they would ever get in measuring the chronological age of a patient. We've gotten this down now below 2. And so they're getting even better the more we control for those immune cell subsets. And so what we're essentially finding is that as a feature for any algorithm that uses epigenetic methylation is a good thing because it makes your results and your predictions much more accurate. And so those two things are sort of involved together, both the multiomic clock that we're doing as well as this new feature method and this new immune deconvolution method. But those are probably the papers I'm most excited about because eventually what they're doing is creating better and more accurate and more robust epigenetic clocks, which can tell us more about that aging process, which still, even though we're doing a lot of other things, is still our number one focus because of its importance to everyone's daily life.
Yeah. It's cool to see you guys actually actively involved in the research, not just creating the infrastructure behind it, but also like helping the researchers and collaborating with them and progressing the industry.
It's something we set out to do from day one. And the reason being is actually more for our benefit than theirs is because, we can obviously help in terms of processing and validation samples, but they've been able to help, I think, in terms of how to do this research and communicating all these really great and important lessons. And so our collaborators have been amazing. We're always looking for more as we've got now a lot of data that will take us years and years to analyze by ourselves. And so we're really trying to leverage that and make this epigenetic methylation search really a really tight-knit community, trying to improve on predictions, not just for age, but of heart disease, of diabetes, of all of these different outputs.
Well, Ryan, I want to switch gears now and talk about some of the interventions, the things people can do or research today to start making a difference in their biological age and then also their performance.
Yeah, certainly. And so, this is at the end of the day, the question everyone wants to ask is, I've taken my test. Now, what do I do about it? So happy to pull that thread a little bit, talk about, I think, what we see to be really helpful and what we're excited about in the future.
Yeah. So I think everyone knows that the first place to start, they might know, but it's difficult to implement, are the lifestyle basics, such as getting out and moving, sleeping, eating right, these types of fundamental basic things, stress management. And like, what else do you see beyond that? Or are there any nuances there that you think need more clarification?
Yeah, I think that surprisingly, one of the biggest nuances there relates to activity, physical activity, and in these large meta-analyses, the relative weight of this the impact of physical activity tends to be a lot smaller than things like, for instance, BMI, which we obviously know that those two things are highly correlated, and so the question might be we always wanted to know is maybe why is that the case, and we haven't published on this or I would say done any controlled trials, but we are noticing a trend which I think is that those people who work out all the time, who are even obsessive about working out, our Olympians, our professional athletes, they tend to have worse aging than those people who are maybe very similar in other biomarkers, but are not doing that amount of exercise. And so, even though we haven't done a performed study on this, I'm starting to believe that we should do it because I think that some exercise, a moderate level of exercise is great. Even pushing moderate level of exercise is great. But I think that once you get into, very serious, maybe overtraining, you can actually start to have some negative effects of that. And that might be due to things like increasing reactive oxygen species that might be having some negative effects on things like your mitochondria or, your DNA damage type of responses. We're not really quite sure of the mechanism, but I would say that it's pretty clear in our data now that we're starting to see that really, really serious exercise, a lot of stress on the body is not always a good thing. And so I think that's one of the most surprising things that we found as it relates to a lot of those lifestyle features. It was probably a little bit less intuitive than you might imagine.
Yeah. My approach to movement is fairly simple. It's a lot of low level movements, restorative movements, things like walking or qigong or tai chi, or even weighted walking, which is called rucking. There's like so many little things you can do just to get yourself moving, to get nutrients flowing through the body. And then every once in a while, layering on something like whether it's resistance training or it's sprinting, something that's a little more explosive, like higher intensity, stimulates like an anabolic response, but like avoiding constant workout stress, and how often that is will depend on the individual.
That's a really, a really good way to approach it. For instance, I would say muscle health is really important for longevity. There was recently a study that published, grip strength is a major determinant of things like lifespan. And so we know that that's important, but we also know one of the major reasons people die is due to cardiovascular disease. Obviously, people know to combine aerobic and anaerobic, but I think that beyond that, I think that a lot of the things that you're saying, things like mobility related exercises, they also tend to have a really great improvement, especially if you can combine them with, more, I would say, mindfulness and things of that nature. And I think that goes to the other big surprise. I talked about on the first podcast how things like meditation has been a very big surprise for us. But even since then, there have been a lot of studies that have come out that show, for instance, things like work stress. For instance, for anybody who works more than 40 hours a week is on average 1.5 years older than people who do not. And things like emotional regulation, you can see these trajectories. The worse you are at regulating your own emotions, the more you're going to age. And so I think we're starting to see now this body of evidence really come that stress and the ability to handle your own stress internally are really big drivers. And so I think if you combine a lot of those mobility related exercises with some of that, sort of stress maintenance at the same time, being mindful about your body, how you're moving, and all those different things all coalesce into some really great benefits for biological aging.
Absolutely. If you were to take any of the typically seen as pillar areas of health, wellness, performance to focus on, which would you say is like the lead domino that you should focus and prioritize this first and everything else becomes easier?
Yeah, well, I would say it's probably no surprise that it's going to probably be metabolic health. I think that insulin resistance is something that can hurt your body in numerous ways because of those downstream cascades. And so I think that however you would like to handle your metabolic health, whether it is through diet or exercise or whatever your approach is, I think that that's going to be the number one outcome. Which again is why we see BMI having such a big correlative to all of these different clocks. And so I would say that definitely controlling metabolic health is very, very important. And I think the ways that you do that can be diverse, and they all seem to be important.
Mm, nice. Okay. Well, I'll list a bunch of different like molecules and things to take and do, and you can give me your thoughts. And since there'll be a lot of them, it doesn't need to be a long answer. It can be just a 30, 40 word answer or yay, nay, however you want to respond, but just so that people can get some thoughts from someone involved in the industry on all these different things.
Certainly. And I want to preface it by saying there might be a lot that I say I do not know, because I do not want to offer any guidance in something I'm not quite certain about myself.
Absolutely. And these can be both for longevity or performance.
Yeah, certainly.
All right. The first one is methylene blue.
Oh, I like Methylene Blue, particularly for performance and for anti-aging. I think that its ability to help with mitochondrial dysfunction can be great. And I think, again, you're looking
Yeah.
at, as we talked about metabolic health being an underlying process, mitochondria as an underlying aging process is obviously huge. So I like Methylene Blue.
Okay. What about the antioxidants that can penetrate the inner membrane of the mitochondria, such as melatonin, glutathione, superoxide dismutase, and chlorophyll?
I think out of all of those, I'm probably most qualified to speak on the glutathione, which we do see to improve biological aging criteria. I think that glutathione and detox pathways like the N-acetylcysteine, I think that those things tend to have health span benefits in large cohorts, and I think in our testing they have mild but positive benefits.
Okay. What about the senolytic agents?
Yeah, so this is something that's recently changed for me. I'm still a believer that senolytics are exciting and should absolutely be developed, but I think that we're about to, I would say, publish it'll probably be, I would say, February or March, but we're about to publish a study on dasatinib and quercetin, the combo of pretty hard chemotherapy drug, quercetin, which probably everyone's familiar with, have been really well documented as senolytic, but we did not necessarily see age improvements with our testing. We actually saw age worsening in a majority of patients. And so it was only 19 patients, so it was very limited in its total number. We're already doing a follow-up. So we're doing a follow-up to include another, sort of herbal senolytic in the fisetin to have more data there. But I think that there have been other data sets coming out showing that just targeting senescent cells in really high volumes without specificity might lead to negative effects instead of some of those positive effects. And so I think that, it's too early to say with that data set, but I think that I'm maybe a little bit more skeptical about their widespread use than I was previously.
Yeah. And I think that there's like two main camps. The one that you take it a small dose every day and one that you take it a large dose like once a month ish. I'm more bullish on the once a month style because I think that they also are signaling molecules in a way, and they can have like benefits in the right doses at the right time. And if you're indiscriminately suppressing them every day, even if it's only a subset of them, I don't think that's the more biocompatible approach.
That's the approach we're taking with our newest fisetin study. That's one we actually adapted to try and see if it made a difference. And so certainly we can talk about it hopefully next time.
Okay. What about pterostilbene?
Yeah, so Pterostilbene, usually goes hand in hand with resveratrol. A variety of things, as SERT mimetics is, activating a lot of those sirtuins has been one of them. And I think that the data doesn't look as promising there as I used to think as well. A lot of backlash for, so I think some of that sirtuin science, which I think is well founded. But I would also say that from our perspective, we don't see a ton of changes in the markers that we test.
Mm-hmm. Okay. Spermidine?
I don't know about spermidine, and I wish I did. I wish that we would do a study on this to look at autophagy and spermidine. I would be hopeful for it, but unfortunately, I don't have enough, I would say, experience with it myself or with our testing to really know in one way or another.
Okay. What about astaxanthin?
That one again looks to be pretty positive. I would say astaxanthin and a lot of those plant molecules, for instance, even things like the carotenoids are from things like carrots that people might be familiar with. So some of those pigmented molecules do seem to have associations and effects. And so again, I would view that one positively.
Okay. And something very basic that is like very accessible to most people, vitamin C.
Yeah, so vitamin C is actually surprisingly a demethylator at really high concentrations and volume, and so this is, again, I don't feel qualified to speak on it yet because it's acting almost directly as an epigenetic regulator, and we haven't done a lot of studies on high dose vitamin C or low dose vitamin C. I think that though phenotypically in most of the studies, I don't see a reason not to do it.
Okay. And then we'll go on to the blood sugar regulators, the metformins, the berberines, the rock lotuses, the certain vitamins and minerals. How do you feel about those?
I don't even know about rock lotus, so I'm making a little bit of a note to follow up on that. But in terms of berberine, I think again I feel very positive about the berberine, even the dihydroberberine, one of those newer, maybe more available bioavailable molecules. I think for metformin, I still like the metformin for certain people. But we have not seen maybe some of the longevity related benefits in epigenetics that I think a lot of people are claiming. And so I'm really excited to see things like the TAME trial, this large study of metformin, come out with a multitude of biomarkers to really judge its efficacy. But I'm hopeful, but again, a little bit more skeptical recently.
Yep, that's actually exactly what I'd say. I'd say that metformin's the one I'm most skeptical of that on that list, but time will tell whether it's as good as the initial research stated and if there's any other side effects we don't yet know about.
Certainly, completely agree.
Yeah, okay. Let's go on to okay, we can't forget the lifestyle things. How about like getting outside and getting sunlight? Like that's a controversial topic in longevity for some reason.
Yeah, the reason I would say it's probably not as controversial from my perspective is because we know how great vitamin D is. Every one of these published clocks, the Horvath, Hannum, PhenoAge, GrimAge, Judith, and Pace, all show positive associations with more vitamin D. And so how it relates to maybe other, aspects of sunlight, like circadian rhythm regulation, or how it relates to, UV exposure, which might be a bad thing, I'm not quite sure, but I think that any process that encourages vitamin D is certainly going to have a positive impact on the aging process.
Yeah, I think part of it's when you get that sunlight exposure. If you get it in the early morning and then a little towards sunset and a couple minutes at solar noon, that'll give you like that'll entrain your circadian clock. And we also know that exposure to sunlight is important to get some UVB, which sulfates and activates vitamin D. It's D isn't activated in the same way, so if you can overdose on it, you can have all kinds of issues and complications from taking a ton that's not actually activated.
There are studies going on now that we're helping with about the time of day methylation measurements with some of those behaviors, because I think a lot of people do believe that the circadian rhythm is incredibly important to health, but maybe even as a confounding factor like those immune cells.
Mm-hmm. And then what about red light therapy?
This is another one that I don't know. A lot of conflicting data, I think, and nothing that I would say unique data on. I think from a skin perspective, I've probably anecdotally seen some very positive effects, but outside of the general whole health, I'm not so sure.
Do you do anything to filter your water?
Yeah, definitely, and I would say that we do sort of a couple different purification systems here at our house. I think it's important, as we talked about, for some of those chemicals that we might be getting a lot more exposed to but don't know about. The more pure your water, filtering microplastics or any of those other things, I think is definitely a good thing.
Okay, what about any other essential like lifestyle things you can do?
I think that if you're generally moving, getting exercise, getting sun, I think if you're doing those things, that it's all great. I would just mention two things that keep popping up in the epigenetic literature, which are a little bit unique, and that is exposure to green space. Green space. And I'm not even sure how to quantify that. I'm not sure if it's the lack of pollution or P2M or what is having an effect, but there is no denying that we see green space having a positive effect on a variety of markers. We can even look at zip codes and how much green space there are on the zip codes and look at really broad biological aging trends due to the exposure to green space. We've even seen that IQ changes happen in children so better IQs for those people who are exposed to more green space. And again, I'm not sure what might be mitigating that or mediating that effect, but there is an effect there.
Okay, one thing I forgot to ask you about is the whole controversy over NAD boosters and NMN versus MIB626, the new patented version of NMN. Where do you stand on that?
We see, I would say, very mild but slightly positive effects on epigenetic aging with some NAD boosters, and I think it might relate to your level of deficiency or age or health status in terms of how you're seeing those effects. I think that I used to be a big proponent of more of the NMN. I'm sorry to hear that the FDA might be taking it away. Like the N-acetylcysteine, I was a big fan of the N-acetylcysteine and still am. But I think that the nicotinamide riboside looks to be staying around, and it looks to have a lot of positive health benefits. So I think that it's definitely something to look at. At this point, though, we've done some experimental testing with, even just niacin, regular, B6, in terms of how it's relating to those NAD levels, and we're seeing, relatively positive benefits. There are a couple studies that have been published, from Finland, even looking at, mitochondrial deficient individuals who can correct those mitochondrial deficiencies just with B6. And so I'm not sure you might need one of those precursors directly, but I think that if I had to pick one, I think the nicotinamide riboside has plenty of positive data.
Mm-hmm. Yeah, I mean, I wrote an article a long time ago about the free and cheap things you can do to optimize the salvage pathway and keep your NAD plus levels high. And there's a lot of other things you can do that aren't super expensive and they aren't flashy new molecules, but
I could totally agree, and I think that we might find, in some of these comparison cohorts, you might see very similar intracellular NAD levels with a lot of those treatments.
mm-hmm. All right. Then on the performance side, are there any small molecules or other things that you find interesting that you've personally experimented with? I have a couple I want to ask you about, but I'm curious about from your own experience.
Yeah, well, as we're talking about these NAD, the 5-amino 1-MQ, I still have as one of my favorite performance enhancing molecules, and actually repair and recovery molecules as well. And I know that there are a lot of studies in pharmaceutical companies now with second or third generation of these, which are even better, but are a little bit less accessible. So I tend to be a very big fan of the 5-amino 1-MQ. I like it for my own athletic performance, probably more than anything. But I also think that if it's increasing my NAD, probably great. I love that NAD plus the NADH ratio go up as well.
Yeah, that's actually one I had on my list for you about performance enhancement. Do you take any nootropics or things for cognitive performance?
I do. So I experiment a little bit. I experiment with like histamine mimetics like pitolisant, the 9-MBC. I'm a regular in believer of creatine. It's, just regular creatine monohydrate for mental performance as well as athletic performance. So I'll experiment with some of those. I also will do a lot of the peptides, probably one of my favorite peptides, which I unfortunately can no longer get. So I hesitate to bring it up, but FGLL was an amazing product for my own memory. I loved it. I think that people will use dihexa. I don't tend to be as big of a dihexa fan from a peptide perspective, but I still think it's great, especially in the long term. So do a lot of those different things. Alpha GPC, definitively like, even the huperazine, rhodiola, I tend to have positive effects with. I won't do them, I would say, regularly, but I think that I do have positive effects. I think so. I play around with a lot of that, but I think that generally things like the basics, like the creatine, tend to work best for me.
Yeah. I have a handful of those sitting around. What about CMAX?
CMAX, yes, another one that I really like. My only issue with CMAX is the duration. Having to do multiple administrations, but other than that, I love the CMAX. I love the C-Link, probably even a little bit more. I can tend to be a little bit anxious, and I think the combo of the two tends to work really, really well for me. I would say, if I have a paper to crank out or writing to crank out, that's when I would start to think about those. Is it sort of it doesn't get me too stressed on it, but allows me to get a lot of work and concentration done.
Yeah, and I have probably 1.5 grams of dihexa lying around. Why are you not a fan of it? I have to know.
So I do. I love the dihexa. I think that with it being 10 million times more potent than BDNF, I think that it's almost impossible not to have a neuro protective or neuro benefit there. The issue is I don't know about the dosing. I don't know about the duration. And so I think that a lot of the dosing out there has been relatively arbitrarily decided on. I think that, but one of my favorite videos to show people is that dihexa video after induced Parkinson's in mice, where I don't know if you've seen it, but it's jaw dropping, right? They're hanging on this clothesline, and you see that the Parkinson's induced mouse drops immediately. Then you just have the Parkinson's treated mouse with dihexa, and then the regular mouse, and they're obviously, with motor function issue like Parkinson's, just trying to test their motor function and how well they can hang up there. The Parkinson's induced mouse that was treated with dihexa hangs up there almost double even that of the normal mouse. And it's incredible to watch. I mean, the thing's basically doing pull-ups up there, it looks like. So, I think that, that speaks a lot to its ability to help repair damage in the brain. And beyond that, I think stimulate, just sort of neurotrophic processes. And so I would say a big fan of dihexa. I just wish I knew how to use it a little bit better.
Yeah, I'm with you there on that front. And also, my other hesitation around it is since it's such a potent activator of neurogenesis and forming new neural connections in the brain and everything, I'm wondering like when to strategically use it. Cause I want to use it when I'm actually like really challenging myself. And I don't want to use it when I'm like traveling or in an unstable environment where it's like a potential to like wire a pathway I don't necessarily want to wire.
Yeah, doing a lot of the research when we were first using it at TaylorMade Compounding, I also don't know when to take it in terms of time of day. I think that there's some evidence that might suggest taking it earlier in the day is better, and then there's also I think some evidence to say that maybe at nighttime is better. And the relationship to BDNF to some of those metabolic processes as well, I think is interesting in that whole circadian, when to take it pathway. And so again, I don't know what to generally recommend there, but I've experimented with both.
Yeah. And then one, it's like a hybrid of, I guess, of body composition and also has nootropic effects, and that is called tesofensine.
hmm Yeah, so tesofensine is one that we again started right at TaylorMade when we were going there. Got a long history, but I do like it. I like it a lot, particularly for weight loss. I liked it better before the sort of I would say mass appeal of these glucagon-like peptides, like the semaglutide or the tirzepatide, which is out now. Before that, it was one of the best ways to lose weight via appetite reduction, but you're increasing epinephrine, you're increasing noradrenaline, so you're hitting this appetite and energy pathways in several different ways. You're increasing metabolic rate, you're reducing appetite, you're increasing satiety. So all of these different combinations, which are great, without maybe some of the side effects that you would get with maybe some others type of stimulants. And so I always liked it for weight loss, maybe a little bit less now. But I think a lot of people also report that a lot of those neurological related focus effects too, because again, you're regulating those same dopamine, noradrenaline, type of transporters. And strangely enough, there's actually a paper out there that says, and I hesitate to say this, but at really high doses, it can actually have even illicit drug-like effects like MDMA and some other things. You have to take it at very high doses. I only say that because with a lot of people probably hearing a lot of those neurotransmitter-related things, they might think, hey, is this addictive? And they've shown that it's not it's significantly less addictive than things like amphetamines, like Adderall.
Yeah, well, of course, we don't recommend using it for that kind of recreational
Yes,
purpose, but
absolutely,
that's
not at
the
all.
data's out there.
I just thought, yeah, I thought it was interesting that at high doses, those neurotransmitters could go in a different way. But yeah, certainly would not recommend that.
Well, Ryan, this has been really fun chatting with you and catching up. And if people are interested in connecting with you or TruDiagnostic, I know we've set up a code for them, but how do they go about connecting with you?
Yeah, so they can always go onto our website. We have sort of a contact us option, or they can email me directly at ryan@trudiagnostic.com. And always happy to share information and data as we have it. And hopefully you'll be seeing a lot of us in the literature here as we got a couple really exciting papers coming out.
Yeah, and my code's Urban. So if you use that, that'll save you on, I believe, all of their testing options.
Yeah, definitely. We have two now. Actually, we launched one since the last test, which is a lower price version and really the only third generation clock available in the Genome pace.
Very nice. Okay, we will go on to your top teachers, and then we'll do a quick rapid fire round and call it a day.
Yeah, sounds great.
Yeah. Okay. So, what three teachers have had the biggest impact on your life and work?
Yeah, it's tough. I think that I don't know that my answers have changed a ton since we last talked, but I still think that probably some philosophers like John Mill. I think I referenced last time. I think a big contributor. I think more recently, I have to throw props out there to a couple of different researchers in the epigenetic space, particularly another Jonathan Mill, who's at Exeter, doing a lot of really great brain research in epigenetics and creating some really cool new techniques. Throw those out there is probably two important people currently.
Cool. What's been your biggest fascination over the last 8 months?
Oh yeah, I think that my biggest fascination has definitively been that imprintome, as we've talked about earlier, just because it's so new and I think the potential is so large.
What's one thing that you've changed your mind on in a major way since then?
Ooh, that's tough too. I think we probably talked about a couple. I think that maybe some of my changing thoughts on some of those sirtuin pathways, maybe over the NAD precursors compared to things like B6. I think those are all starting to change. I don't think anything has changed in a major way outside of that, and maybe that means I need to get myself out there a little bit more.
Okay, what's been your most gifted purchase, or I guess most gifted purchase for anyone interested in your work in this space that's under $500?
As Christmas is coming up, one that I frequently recommend is actually a cold pressed olive oil called Costarina, which has get a lot of great polyphenols. Has been linked to maybe some longevity related things. It's a great gift. It is delicious. It's fun to cook with. So I love that. Very very cheap for the holidays. But beyond that, I would say that. I think that I've started to get a little bit more into even some skincare related things and some skincare testing. There are a couple of companies out there who are starting to do some skin aging related things. I know when we talk about longevity, it's hard to separate the two, but I think that hopefully we'll be announcing sort of a partnership coming up here soon for some skin longevity testing that might even make some specific epigenetic related referrals to different products, which I'm really excited about too.
Wow, that's very interesting. Yeah, I didn't realize the connection between the skin and longevity as much as I should have until a couple of weeks ago when I interviewed Young Goose in the podcast and we talked about senescent cell burden in the skin and all kinds of things like that a lot of people probably haven't never considered.
Yeah, certainly. And in terms of bringing a lot of my former world peptides and the skin longevity, there's another great company out there called OneSkin that does a cosmetic senescent product that is shown to be better than the topical rapamycin for senescence. And so might something to consider there too.
Well, Ryan, any final thoughts, ways you'd like to conclude our episode together today?
No, I think that hopefully, if you have any other questions on this type of testing that we do and the implication of epigenetic research into health, certainly feel free to reach out. But I hope that everyone takes away, some of those really big metrics on how to validate this testing, apples to apples, so to speak, and really dig into those key metrics like hazard ratios and precision. I think that hopefully those are the biggest takeaways for anyone listening, and always happy to explain or provide data to anyone who's interested in that.
Well, Ryan Smith, it's been a pleasure hosting you today. It's fun to explore some of these rabbit holes with you. And I look forward to staying in touch and having another chat like this down the line once there's been some more updates.
Yeah, thanks so much for having me, Nick.